Rokop, and I'm the director of the Broad Institute's Educational Outreach Program. And it gives me great pleasure to introduce our speaker tonight, Dr. Robert Gould. Robert serves as the director of the Novel Therapeutics Platform here at the Broad Institute. He received his BA from Spring Arbor College and his PhD from the University of Iowa. Before joining the Broad, Robert worked at Merck Research Labs, where he served as the vice president both of licensing and external research, and also vice president of basic research. Tonight he will be speaking to us about how our knowledge of genetics and genomics may be changing the process of drug discovery in the future. Please join me in welcoming our speaker, Dr. Robert Gould.
Thanks, Megan. Well, it's a real pleasure for me to share with you some of the processes that go on from when somebody discovers a chemical to when it actually becomes a drug that has the potential to save human lives. And at the end, talk about how things that are going on within the Broad and other institutions around Boston and around the world are affecting how we can do that process more efficiently. So now that my computer's gone to screensaver, let's hope it comes back on. Of course not. So this is the outline of what I'm going to be talking about. I'm going to level set a little bit about what makes a drug, talk about the stages of drug discovery and development, talk about where drugs could come from, what the costs and risks are that are associated with drug discovery, and with that background, talk about some of the opportunities moving forward to change the process that's gone on for scores, if not hundreds of years, and hopefully make it more directed, more individually oriented, and more efficient. So this is a definition of a drug that's taken from the FDA, the Food and Drug Administration website. And basically what it says is that drugs fall into two categories. Something that's useful for diagnosis, cure, mitigation, treatment, or prevention of disease, or anything other than food that affects structure or function of the body of man or other animals. So that's a pretty general description of what a drug means. But with that general description, you can imagine that different ways in which chemicals might become a drug. There's sort of an empirical method. These happen to be botanical products, all of which through empiricism have become drugs. And some of you may be familiar with some of these drugs, hopefully not all of them, since some of them are not legal drugs. But in fact, that's an empirical method of identifying a chemical that changes a structure, or in all of these cases, the function of an organ within the human body. There's actually also lore about how chemicals might become drugs. This happens to be taken from Shakespeare's play, A Midsummer Night's Dream, in which Oberon, one of the protagonists in the play, is describing how he's going to drip into the eyes of Titania an elixir from a flower that we happen to call the pansy, so that she falls in love with the first person that she sees. Now that's another example of, I think, lore, never having actually tried the experiment myself, that extract of pansies will make you change the function of perhaps your brain, perhaps your heart, perhaps your eyes, to change the function of it such that you fall in love with the first person that you see. And from those sort of little examples and from your own experience, you know that drugs, in fact, can then be many, many different kinds of chemicals. They can be natural products, some of the examples I've shown you. They can actually be proteins. They can be small organic chemicals that have been synthesized by chemists. They can actually be nucleic acids, like make up your DNA or within your cells, or the RNA within your cells. Or they can be fats, like lipids. The reality is that any article, using the FDA definition, or any chemical, actually has the potential to be a drug. And so how do we go through the process of realizing that potential to make a useful drug? And that's what we're going to be talking about. Some of the newer things that we actually aren't going to be talking about are that you can make articles, substances, that interfere with the RNAs that are within each one of our cells. And you can see here some companies and some identified programs and some therapeutic indications in which at least 15 compounds or articles are in development that affect the level of RNA within within a cell to affect the function of that cell. Each of those things actually technically aren't drugs yet, because they're all still in clinical testing. And in order to actually be called a drug, in our modern vernacular and with the regulatory agencies, a molecule becomes a drug when it's been proven to be useful. And in order for something to be useful, there's sort of two sides to that coin. It actually has to work, it has to be effective, and it has to be relatively safe relative to the disease that you're trying to treat. And that's a little bit of a moving boundary, if you will. So something might be safe for cancer if the outcome is death, that might not be safe, for example, for something like osteoporosis, where you're going to be taking the drug for many, many years or decades. So the ratio between, or the balance between effectiveness and safety has to be constantly evaluated. But beyond that, government regulatory agencies must agree that it is effective and safe in order for it to be made available to the population.
What other agencies besides the FDA?
Each country has its own regulatory agency. In the US, it is the FDA. Depending on the country in which you are residing, it has its own governmental agency that serves the same function as the FDA. The names are different in different, there's one called EMEA, which happens to be in Europe. China is called SFDA for Sino Federal Drug Administration. But each country, in essence, has its own agency. And in each country, you have to get individual approval in each country from that regulatory agency.
There isn't one for the whole EU.
EU is a little bit different in that they have a mix of a centralized one and then local country ones. So they get recommendations from the central agency that then the individual country responds to and typically agrees with the central agency. Historically, they've been very individual. So if you wanted to sell in Liechtenstein, you had to go to the Liechtenstein equivalent of the FDA. If you wanted to sell in the UK, you had to go to the UK equivalent of the FDA. But they've gone to a more centralized agency. The exception to that, of course, are things that are classified dietary supplements, like herbal remedies, which have to neither prove efficacy nor safety, because they are considered dietary supplements and not drugs. So in the process of moving from a chemical to an actual drug, there's 5 stages that a chemical would go through. And we're going to spend some time describing each of those stages so you have a little bit of an understanding, hopefully, of the science and the information that's gained at each of those stages. So we're going to begin down here in the bottom left corner with identifying a biochemical target that you want to affect and discovery of a compound that affects that target. Move through a stage before you get into human beings that's called preclinical drug development. Move into human clinical trials where the effectiveness and safety are evaluated, but in fact in the other order. Safety is always evaluated first and then effectiveness after that. In parallel, in order to converge at the human clinical trials, there has to be a massive manufacturing undertaking. So imagine having to make thousands of pounds of a chemical substance that's greater than 99.5% pure. And that's the manufacturing challenge that has to converge at the stage of human clinical trials. At that point, what's called a marketing application is filed. That's actually the approval asking approval from the government agency to market the drug. So those are the 5 stages that we're going to be walking through this evening. And as I've said, at the human clinical trials is where safety and efficacy occurs. At the marketing application is where one seeks agreement from the government regulatory agencies that safety and efficacy are achieved. But throughout that, 4 of those steps, regulatory compliance is required. That is, the government regulates many of the steps that occur at these 4. And they have some acronyms that are associated with different stages of that. So at the preclinical drug development stage, everything has to be done under a massive set of government regulations that are called good laboratory practices. At the human stage, it has to be done under a similarly large volume of good clinical practices. The manufacturing stage, it has to be done under good manufacturing processes. And of course, the application itself is reviewed by the regulatory agencies. So in fact, 4 out of the 5 stages, one has to comply with governmental regulations. So we're actually going to begin at the lower left corner of those. And if you ever get lost where we are in the talk, you can hopefully remember this little, look up in the corner here and it'll sort of orient you where we are in those 5 stages. In that first stage of target and compound discovery, you can actually break that up into 4 additional stages. What is the biochemical target for which one wants to achieve a chemical that hits? How do you know that that's a valid target, that's actually going to do something useful? How do you find a chemical that hits that target? And then how do you optimize that chemical for human use? So target identification can actually come from one or more of multiple ways. It can come from identifying and defining medical needs. So, for instance, there's a huge medical need for diseases like Alzheimer's, since there's nothing that's very effective for that. It can come from research on disease mechanisms, so an understanding of what happens when an organ begins to fail, like the kidney or the brain or the bones. Or it can come from an understanding of a biochemical target, that is a protein or some other piece of chemistry within the cell that is associated with either the medical need or the disease mechanisms. And really, starting from the medical need, historically, there's been a great deal of effort of saying, well, here's a medical need. What are the mechanisms that underlie that need? Or what are some biochemical targets that might underlie that need? And that's been kind of the traditional way of doing it. And so there's tools that are available to do that. One can use disease populations, identify genes, proteins, or cellular pathways that are relevant to the disease. So look in a normal person and a person that might be suffering from the disorder and say what's different about proteins in their plasma, what's different about their cells, what's different about their bodily function. You can take another approach is you might take a known drug that actually works pretty well and might be limited either in its safety or its efficacy, and then say, well, if I understand how this known drug works or doesn't work in terms of safety, I can improve it by just simply understanding more about its mechanism. Or you might begin to apply molecular differences. So for instance, if you were interested in what is difference between a tumor cell and a normal cell that occurs right next to the cancer cell, you might characterize the molecular differences between that cancer cell and the normal cell that sits right next to it. So all of these, from epidemiological studies of broad populations, from knowledge that we already have and might be able to improve, or understanding of molecular changes, are ways of identifying useful targets. I'm going to be following just a paradigm to illustrate this for you through the rest of the talk, and that paradigm is going to be around atherosclerosis and the development of a class of drugs that are called statins that lower cholesterol. So in 1948, a large study was started not too far from here in Framingham that showed there was an association between cholesterol levels and coronary risk. About a decade later, a biochemical target was identified, an enzyme called HMG-CoA reductase. This enzyme actually is an important enzyme in the biosynthesis of cholesterol. So here's a population-based study that said, oh, cholesterol is associated with coronary risk. Here's a potential biochemical target that is important in production of cholesterol. And so you can imagine asking questions like, if I stop the activity of this enzyme, can I stop cholesterol biosynthesis and lower the cholesterol that's present in the plasma? That's an example of moving from epidemiology, population-based studies, through biochemistry to a potential target. So that kind of epidemiology study is now actually being transferred in the world today to looking at how genetics can potentially alter what we're looking at in terms of drug targets. So statins and HMG-CoA reductase are one example. And here I've just listed about 8 different drugs that have been associated, that have come from an understanding of genetics. So one is Proscar, which is used for the treatment of BPH, or enlargement of a prostate in men. Actually, genetics enabled that understanding of how to make Proscar because another enzyme called 5-alpha reductase is critically important for the production of testosterone. There's a group of people who are deficient in this enzyme, and among other things, their prostates don't grow very large at all. So that enabled one to ask the question, well, if I make an inhibitor of this enzyme, can I stop the growth, the abnormal hyperplasia of the prostate? Gleevec and leukemia were derived from an understanding of an aberrant protein that was causing the cancer. In osteoporosis, there's an enzyme called cathepsin K. People that have a deficiency in this enzyme actually have a benefit. They have extraordinarily strong bones. In fact, their bones are very difficult to break. So that led to a program in which an inhibitor of this enzyme is not quite proven, but is in late-stage clinical studies to see if it can, in normal older people, make their bones stronger. In the genetic experiment, people that have a deficiency in this enzyme do have stronger bone. Protein called HER2 for Herceptin. And these last four are just very early ones, some of which you may have read in the newspaper recently. And I forgot to, I'm giving full credit to Mark Daly, but I forgot to. I wasn't sure if the recent report that you may have seen in the Boston Globe and some of the scientific literature was actually going to be out yet. This speaks to understanding the genetics that are associated, among other things, with inflammatory bowel disease and some of the GI dysfunction that Mark has done using population-based studies. So genetics can give you biochemical targets that one can then ask the question, if I affect that target, can I achieve a benefit? Well, that's identifying a target, but how do you know if it's a good target? Sometimes it's kind of easier. So you can understand what the function of the target is. You can understand the relationship between inhibiting an enzyme that makes cholesterol and lowering cholesterol. Sometimes you might want to know what pathways does the target regulate. So in the case of the cathepsin K example in osteoporosis, it's not immediately obvious how inhibiting that particular enzyme results in stronger bones. You can ask questions about how important is the pathway in the disease. Sometimes you can say, well, I don't know what the pathway is in the case of cathepsin K, but I know it's important because people who are aberrant in that enzyme have really strong bones. So it must be important even though I don't know what it is. And then part of validating the target or believing that it's worth working on really gets back to the balance that I made, balance between safety and efficacy, the point I made earlier. That is, if a drug interacts effectively with its target, but at that dose causes an unacceptable side effect, then in fact, the efficacy is tainted, if you will, by the safety profile. That is, you have to always balance what the risk is associated with whatever drug you're using. It's just like many of you, and maybe all of you, drink your 8 glasses of water a day or aspire to drink 8 glasses of water a day. That's pretty good. If you were drinking 80 or 800 glasses of water a day, you would actually exceed the therapeutic index for water. You would have moved from an efficacy point in which water is good and effective for you to remain hydrated to a point in which you had an unacceptable side effect, which is you can actually kill yourself by drinking too much water. Now, it's a massive therapeutic index in that case, but nonetheless, even something as benign as water has a therapeutic index. So we need to understand what that index is as we begin to validate a target. There may be a target that's so important for some function in life that you cannot, in fact, inhibit it safely without having an intolerable side effect. So, in fact, this target validation, staying with the development of statins, I'm now going to change to this kind of timeline to bring you from the 1940s and 50s that I've already talked about all the way up to 2008. And we're going to be sort of populating this timeline as we go along to illustrate some of the points. So in fact, inhibiting HMG-CoA reductase, the enzyme that was identified in the 1950s as being important in cholesterol, in fact, truly wasn't validated until the mid-90s, in which it was first clinically demonstrated that, in fact, if you reduced cholesterol, you, in fact, would reduce mortality. That, in fact, made this connection that was first hypothesized in the 40s and the 50s, actually made it strong. It took until the mid-90s to do that. That's a validated target. You inhibit the enzyme, you lower cholesterol, people don't die. That's pretty valid as a target. To get to that point, the drug had to be approved. Remember, we talked about the fact that the agency has to agree with you. In fact, from the time the agency agreed that a compound, lovastatin, was effective, it lowered cholesterol, was safe enough to market to people, until it was actually proven to reduce mortality, was about a 7-year period. So this timeline can actually become very, very long in validating a target. And that's one of the ways in which our understanding of the human genome has the potential to have a profound effect by decreasing these decade-long gaps between various important stages, because we'll understand more about what the human body is doing. Cholesterol, so I've drawn the distinction in the last slide between lowering cholesterol and saving human lives. Lowering cholesterol is an example of something called a pharmacodynamic assay. Pharmaco means drugs, so pharmacology. Dynamic is what it sounds like, dynamic. So it's the active effect of a drug. And it's actually a measurement that reflects something of the chemical something that the chemical does when it's put into an animal or a person. So in the example that we're using, lowering cholesterol, in fact, is a good thing, but it's a pharmacodynamic endpoint. What you really want to do is stop people from dying. That's the real point. But lowering cholesterol enables you to get there through this pharmacodynamic measurement. Pharmacodynamic measurements are important because you can use them in preclinical animal work to say, am I affecting a target? And then you can carry over that understanding from animal work to your initial clinical trials. So you can say, for example, what dose of lovastatin, in the example we're using, lowers cholesterol. The cholesterol is a pharmacodynamic assay. In the preclinical animal studies, both biochemical and biological, it was known that lovastatin inhibited this enzyme and thereby lowered cholesterol. And so by connecting those two, you can begin to do a better job of picking your doses in people. Again, picking doses using the water example is critically important. A dose of water that is equivalent to 8 glasses a day is safe. A dose of water that's equivalent to a swimming pool a day is not safe. You need to find where you are in your dose range for chemicals, be they water or chemicals that have been synthesized. And pharmacodynamic assays enable you to do that. So we've got a target identified. We've got a target validated in some animal model, perhaps through a pharmacodynamic assay. Now we need to find a chemical that affects that target. In fact, some of those things that I showed you earlier, the traditional medicines and the lore, have actually not been that productive for pharmaceutical companies or for biotech companies for a couple of reasons. Some of them have been extremely effective, but across the board, they've not been overwhelmingly useful for identifying chemicals as leads. And that's primarily because they tend to be complex mixtures of compounds. And so trying to sort out what the active ingredient is in a complex mixture that is an extract from a plant or some other natural source can be impossible. In fact, most drugs, if you just went through a listing of drugs, do come from that. They do come from purified components of natural products. But the important point is that of the drugs that are approved, they're purified components. There's a lot of natural products that are food additives that are used for a variety of things, but they're classified as food additives, not as drugs. So if you look, just staying with our cholesterol example, in fact, lovastatin was approved in 1970. Lovastatin was actually discovered in a fungus, as was the first inhibitor that was ever discovered. These are actually both ingredients were identified as natural products, purified components of a natural product that happen to affect the cholesterol biosynthetic pathway. So this has been a productive source in some examples of drugs. Another way that's been used is what's called modeling approaches. So you know what target you want to hit. You can do computer modeling to say, well, here's this big protein that I want to attach a small chemical to, and you can allow computers to do that kind of modeling approach to tell you how a chemical might interact with that protein component that you're interested in blocking. In fact, this has been most useful when you already have a hint of what something is doing. So you kind of know something's interacting, and now you want to make it better, as opposed to where do I start from, how do I find something that doesn't interact with anything. The computer modeling is less useful for the identification and more useful for the optimization step. In fact, most modern drugs, that is drugs that have been approved in the last few years, actually come from massive chemical libraries at biotech companies and pharmaceutical companies where they screen against large numbers, millions of chemicals to try and find the few that might interact with the target of interest. So hundreds of thousands to millions of chemical compounds might be screened against your identified validated target as you try to find the lead, because robots can do the same thing over and over and over and over and over again, precisely the same. That's often run with robots running these automated assays. Robots don't start thinking about other things in the middle of an assay. They move with extraordinary precision and certainty that what they're doing is precisely correct because somebody's told them it's precisely correct. And they will do that for days on end. However, to do that, you have to manage information that comes from millions of compounds. So if you're thinking about, gee, I'm interested in making drugs, what kinds of jobs might be out there? It actually, at this level of just screening, there's a huge amount of expertise that's required at different steps for that. This happens to be a CAD diagram of our system that's upstairs from here of high throughput screening or robotics. Each of these modules down here that are the robot lines, just to use this one as an example, this one as an example, is about, on this dimension here is about the same size as this screen here. And the vertical dimension is about 9 feet tall. So just to give you some dimensionality on these. We actually have 2 of them that are the same size, and then this one, which is about twice the size of that upstairs. And these robots have robot arms that, this is an example of one of them. There's a robot arm that you can just see sitting here. There's instruments that actually are some of the same instruments you can see out here in the foyer. And the robot arm just moves chemicals and plates around from place to place, hours on end with extraordinary precision and perfection. We have a couple of these systems that can actually, because we're interested in treating infectious diseases, we actually have a couple of these systems that can actually handle viruses and bacteria in this system. So we can attack or test against viral targets or bacterial targets. In fact, we've been talking about using lead identification as a chemical. In fact, you can also have biological leads. That is, leads that come from biology. So RNAs, as I talked about earlier, can be designed to form a complex with nucleic acids within your cells to knock those nucleic acids out and thereby achieve a change in the function of the cell. You might mimic protein sequences, mimicking a hormone like insulin. For those of you in the audience who might be diabetic, in fact, insulin itself is a good identified lead. It hits a validated target. And in fact, there's varieties of insulin, long-acting insulin, short insulins, all of which mimic the protein sequence. You can mimic a sequence of an enzyme like TPA or tissue plasminogen activator that has an active role in the body to dissolve blood clots. Now, if you happen to get a blood clot in a coronary artery and thereby block blood flow to your heart muscle will not get oxygen and you will have a heart attack. You need to get blood flow back to that heart muscle as quickly as possible. So mimicking a sequence of an endogenous or a naturally occurring protein enables you to do that. If you have a genetic disease, you might mimic the enzyme that is deficient and replace it. And more, we're seeing monoclonal antibodies, that is, antibodies that are directed against protein targets and achieve an effect as a way of identifying biological leads. So now we've got a lead. Now we're going to turn to lead optimization. Just to remind you, we're still at stage 1 of this of the 5 stages. You're now about, if you were starting this as a young scientist back here, and now you're at lead optimization, you might be about 2 or 3 years into your project at this point. So you have to be extraordinarily patient if you want to discover drugs. But we're finally at optimizing the lead. And when you optimize the lead, you need to ask questions of potency. How strong is the chemical? It's a good thing to give as little as possible to get the effect. And so you do in fact want to make a chemical that is quite potent or quite strong, so you don't have to give massive amounts. You really want to make a chemical that only interacts with your identified and validated target. You don't want it to hit 20 or 30 or 40 other things that are not relevant, because that inevitably is going to lead to a safety problem. So you need to ask questions of potency, you need to ask questions of selectivity, and you need to ask questions of effectiveness. Once it hits your target, does it give the endpoint that you're interested in? Once your lead hits HMG-CoA reductase, does it lower cholesterol like you hoped it would do? Does it do it potently, and does it only do it on that enzyme? Those are the kinds of questions that one addresses in the lead optimization. In fact, chemicals also need to be optimized to be handled by the human body. We expose ourselves to chemicals all the time, every day. Those of you who took the T over may have exposed yourself to more chemicals recently than you wanted to if you were, stuck down in the green line somewhere. But we're exposed to chemicals all the time, and the human body is very good at handling those chemicals. So our leads have to be optimized. They have to be optimized for absorption. If we're taking a pill or a tablet by mouth, we have to get it absorbed into the body. We have to ask questions about how well is it absorbed. And we want it to be optimized for absorption. If you take a certain amount of a pill, mainly from a cost point of view, it's really useful to have all of that medicine that you take absorbed. If you only absorb 10% of it, then effectively you're flushing 90% of that chemical down the drain. That's just not a cost-effective way for anybody. So you want to get as much absorbed as possible. The other reason you want to get as much absorbed as possible is each of us are different. I might only absorb 10% of a chemical. You might absorb 90% of that same chemical. Effectively, that means you're being exposed to 9 times as much of that chemical as I am. That gives you a huge variation in how much chemical your body is being exposed to. However, if all of us are absorbing it at around 80%, then the biggest it could go to is from 80 to 100%. And so you can reduce the variability by making sure that you optimize the drug to be absorbed by the human body. But once it's absorbed, where does it go? It has to get to the right place. If you get something absorbed through your gut, after the gut and after the blood, the next organ that it's going to see is the liver. So if you take a chemical, it gets absorbed in your gut, goes to the liver, and then the liver immediately does what it's supposed to do, which is get rid of this thing it's never seen before, but you're trying to treat something in the brain. That's not very effective. You have to get it absorbed, and you have to get it to the organ where you want to have its effect. In fact, the brain is a particularly challenging organ in which to get chemicals, because our brains and the brains of all animals, are exquisitely set up to pump chemicals that it's never seen before out of the brain. So a chemical can go into the brain and get pumped right back out with chemical pumps that just are there to get bad things out of your brain. So it has to get to the right place. Our livers in particular, which is why the arrow is pointing at where this guy hopefully has a liver, our livers and other organs, but particularly the liver and the gut, are set up to change chemicals that they've never seen before. There's enzymes in your liver whose job it is to eliminate chemicals that have not been, that your body has never seen before. And so you have to optimize your chemistry to get by the liver and by some of the effects that your body is trying to do. And finally, how is it eliminated from the body? If it goes through the kidney and you have kidney failure, you're not going to be able to eliminate the drug as effectively as somebody with normal kidney function. So you need to optimize how it's eliminated from the body. And finally, you want to know what toxicity does it show. This poor guy took some drug that gave him 4 legs and 2 arms. That's really not an acceptable toxicity for whatever his ailment was. So all of those things go under an acronym that's called ADME-TOX for absorption, distribution, metabolism, excretion, and toxicity. And so you have to optimize for ADME-TOX. All right, we're finally to stage 2, preclinical drug development. We've identified a target, we validated the target, we found a lead, we optimized the lead. Now we really want to get into preclinical drug development. This is the first step that's actually regulated by the government agencies. So it really boils down into 3 main areas. Additional ADME-TOX studies. So those ADME-TOX studies have to be done under those procedures called good laboratory practices. You might do exploratory ADME-TOX beforehand, but now you have to do very precise government-mandated ADME-TOX studies. You want to formulate the drug. So formulate means you want to know, are you going to apply it as a capsule? Are you going to apply it as a tablet? Are you going to apply it as an ointment? Are you going to apply it as eye drops? How are you going to formulate the chemical? And how are you going to deliver the system? Is it going to be an injectable? Is it going to be an oral drug? And then finally, you have to develop chemistry around making the thousands of pounds of the drug that ultimately are going to be needed. And so you need to optimize that chemistry so that it's as cheap and efficient as possible. So toxicology are those studies that are designed to show functional as well as structural changes in a drug. And so you have to, in toxicology study, evaluate how the drug is working. You have to evaluate where it's being administered and you have to evaluate the mode of the toxicology. You have to evaluate the site of the toxicology. Where are the functional and structural changes occurring, and how big are they? Are they related to the dose of the drug? Is there differences between males and females in those effects? And finally, you have to ask questions about latency. That is, if you take a chemical and a toxic effect shows up a year later, that's a long latency. You have to be able to understand those kinds of latencies. You have to understand if it progresses. If you have a toxicological effect that gets worse and progressively worse and worse and worse, that may be more intolerable than an effect that occurs but never gets any worse. And you have to understand if it's reversible. If you can stop a drug and make a toxic effect go away, that's really good. So in fact, this is a case where there's international harmonization on these guidelines. So there is a group called ICH, or International Conference on Harmonization, that in fact is working across those various government agencies to set common global standards for all agencies across the world, actually. And so that's an attempt by the government regulatory agencies to work together to ensure that different countries and different people aren't held hostage by their governments to different degrees and different standards of safety. So back to our statin timeline. Remember Compactin, the first one discovered? About a decade after it was discovered, it was suddenly stopped. Now often when drugs are stopped, nobody knows why they're stopped. But the rumor at the time was that it was stopped for toxicity reasons. In fact, lovastatin, a different drug, was already in clinical development in humans. But when this drug was stopped, the lovastatin trials were also stopped because all of a sudden here was a toxicity effect of an unrelated chemical acting at the same target, and it was unclear as to what that was due to. Now, it turns out that this toxicity happened to be a rat-specific event, so humans were not at risk, but at this point in time, nobody knew that. So the drug, an effective drug that saves lives, was actually stopped for a long time. Well, how do you, rodents and people don't live the same lengths of time. Mice live about 2 years. Humans, might live 80, 90, 100 years. So in fact, how long you're going to do a clinical trial dictates how long animals get exposed. And for safety studies, you have to do a rodent and a non-rodent species. And if you want to do a single dose in humans, you have to treat for about 2 to 4 weeks in animals and show that it's safe for 2 to 4 weeks of repeated dosing in animals to do a single dose in humans. If you want to, and you probably all read this by now, if you want to do more than 6 months of dosing, you have to do about 6 months in rats and 6 to 9 months in non-rats to do a study. Actually, if you're going to market a drug, you also have to do carcinogenicity studies to evaluate whether the drug causes cancer, and that has to be 2-year studies in mice. So that, in fact, prolongs beyond this the length of studies that you would have to do in animals. So now you have to turn to formulation, which are important questions like, are you going to make your pill pink or yellow or blue and orange or white? Are you going to make it a tablet or a capsule? And that's actually not trivial. The entire formulation is the recipe for packaging this. And it's important to get your recipe right for a number of reasons. First of all, so that if you're taking 2 or 5 or 10 drugs, you can look at them and tell the difference between them. If every tablet was a small white pill and you were taking 10 of them, you would have no way of knowing which one, if you took the right one, if you took the wrong one. And so, in fact, the appearance, the shape, the color is actually another government-regulated event, because you don't want two pills to look too much alike. Sometimes they're distinguished by a number stamped on them, but it's better if it's a color and a shape. But that's the recipe. It includes both the active substance and something called an excipient. An excipient is anything other than the active chemical that's added to the pill. So I happen to be virtually dependent upon an antihistamine called loratadine because I appear to be allergic to virtually everything in the universe. And if you look at excipients that are in the Rite Aid loratadine, these are excipients that are in there. None of them have any pharmacological effect. The effect is carried by a chemical called loratadine, but there's cornstarch in there, there's lactose monohydrate in there, there's magnesium stearate in there, there's pre-gelatinized starch in there. All of those things are intentionally added to make the pill that I take. And they're added for a variety of reasons. They're added to increase the size of the tablet. You might only need 10 milligrams of a chemical, but a 10 milligram tablet is really small and really hard to pick up. It's physically hard to do. And as people get older and their fingers get arthritic, it gets even harder to do. So about 100 milligrams is a convenient size. If you only need 1 or 2 milligrams, that means you've got 98 or 99 other stuff in there just to get it to a size you can pick it up. You might want to mask an unpleasant taste. I've already talked about the fact it's useful to identify it. You might add excipients to extend the shelf life so that you can keep it around longer, especially if people are throwing their pills in medicine cabinet where the temperature might go up on a nice hot day where you've taken a hot shower and steam is in there. You want to anticipate shelf life. You might want to control the release of the active ingredient, and you might just make it easier in manufacturing. You're making hundreds of pounds of drugs. There's times where you just want to help the manufacturing process by adding these incipients. So while all this is going on, manufacturing is over here trying to make thousands of kilos of material at high purity and in an economic manner. So just imagine that across the world there's 1 million patients, just to pick an example, that are taking every day 100 milligrams of some drug. If you just multiply that out, that means manufacturing has to make about 36,000 kilos, about 2 pounds, so 70,000 pounds of chemical that is greater than 99.5% pure. That number is regulated by the government every year in order to satisfy that demand. So manufacturing is dealing with this problem of creating massive quantities. All right, we've got drugs developed, we've got them manufactured. This is the first time that you actually now go to the government, depending on what country you're in, and you say, I would like to test this new material in humans. And you have to formally request that. And they take a look at the toxicology that they've done, the manufacturing process. Do you know what the chemical is? Do you know what the other additives are that are in there? And they either say yes or no. You can go into a human clinical trial. This is actually a scary point in the development of a drug. Because all of a sudden you're taking a chemical that who's never been exposed to a human body before, and you're asking people to expose themselves to that chemical intentionally, and you're not exactly sure what's going to happen. And for many of those people, there's zero benefit. It's 100% risk, zero benefit. And so human clinical trials are regulated by a lot of ethical considerations, things like social value. Will an improvement in health or knowledge result? Is it scientifically valid? That is, are there no biases to the study? Are there clear objectives? Have people thought about how many people have to be exposed to get an accurate answer? Subjects have to be selected fairly. They can't be selected based on privilege, convenience, or vulnerability. 30 or 40, 40 plus years ago, prisoners used to be used to do drug testing. You know, that fit because they were convenient. They were there, but they were being selected based on convenience and based on vulnerability. That's not an effective way. That's an unethical way to do a clinical trial. And so you have to define what inclusion criteria, that is, how do you include people and how do you exclude people? Subjects have to be informed of the risks and benefits. They have to be able to stop at any time. There has to be minimal risk and maximal benefit. The only caveat on this one is that the first time a chemical goes into a human being, there's typically zero benefit or unknown benefit and a lot of risk. And so you minimize that by starting with microscopic doses for the first time that you expose somebody. And finally, you have to have independent, impartial bodies that review the trial to ensure that it's being done safely. I'm sorry, not finally. You have to have that. And then finally, you have to provide respect. And if a subject says they want to be removed from a clinical trial, you must honor that. So all of those ethical considerations go into 4 phases of clinical trials. Whoops. The first phase is safety data. How is it being handled? Is it hitting the biochemical target? Is there a result from hitting the target? And really, the purpose of the phase 1 study is safety. If it successfully gets through that, it goes to what's called a phase 2 study, in which more safety is generated, often in the patient population of interest, and then you begin to ask doses. How much do I have to give? You're asking some of the same questions. Is it hitting the target of interest? Is there a biochemical result from hitting that target? But more importantly, what's the minimum effective dose that's needed to get these 2 results? You want to give the minimum dose that's necessary. You get through that, and now you get to large studies, phase 3 studies, that continue to look at safety, but also look at efficacy. Is there a clinical benefit at a dose that you've picked here that hits the target and achieves a biochemical effect? But more importantly, are the risks worth any benefit you might be seeing? And that's the purpose of the phase 3 trials. If you get through those, you get governmental approval at that point. And then you also have to then do phase 4 studies. This is safety in population at large, many, many people. And in the last few years, there have been fairly widely publicized studies of drugs that when they got in, that while they were safe and effective at phase 3, proved to have unacceptable safety once they got into large populations. And as a result, many of those drugs get pulled off the market. There's drugs that have been used to treat diabetes that have failed when they've gotten into population at large. There have been drugs that have been used to treat the pain associated with osteoporosis that have had an unacceptable safety profile when put into large numbers of people. So back to our timeline. Last time we saw it, we had just stopped Capactin for toxicity, and lovastatin had been suspended. In fact, lovastatin trials resumed, but they resumed in patients that had a particular genetic disease, a disease in which they made too much cholesterol. And so the cost benefit there was here's patients who are going to die of cardiac disease at a very young age. Can we treat them safely? And in fact, that's how lovastatin resumed. An earlier drug, triperenol, that lowered cholesterols, was actually effective at a dose that caused cataracts. That was withdrawn because lowering your cholesterol at a dose that causes cataracts is an unacceptable risk-benefit. Here's a drug, cerevastatin, that had an excessive risk of something called rhabdomyolysis when it got into large-scale patient population. In fact, was withdrawn. And if you just jump up to 2008, in fact, it's really not exactly clear what causes this particular muscle degeneration, nor who's at risk for it, nor what patients would benefit most so that you might accept that risk. And because those questions aren't really known, cerevastatin was simply pulled off the market and never put back. Those are the 5 stages. We've walked all the way through from target and compound up through marketing applications and manufacturing. I hope I've given you some examples of the different activities that go on there, some of the regulatory steps that occur at those various stages. And so that's the stages of drug discovery and development. A question that's often asked is, well, good, but we all know, quote unquote, know the drugs actually come from universities and other places, and all that happens is the pharmaceutical companies put them in a nice box and generate cash. And in fact, the question is often posed in various forms as, does the private sector, pharmaceutical companies or biotech companies, actually make any valid scientific contribution to new medicine at all? Or are they simply piggybacking on the hard work of others? In fact, it's been so controversial that people fall on both sides of the coin. This is a study that, here's the website you can get it off of, that you can see what, you perhaps can understand what they're going to conclude. The Truth About Drug Innovation, 35 case studies on private sector contributions to pharmaceutical science. And so what these people did is they reviewed the literature on 32 drug classes, and they ranked 3 kinds of scientific advances. Now, the scientific advances that they ranked were basic science, which roughly corresponds to what I've called target and compound discovery, applied science, which roughly corresponds to these 3 stages, and then clinical improvement, manufacturing, things like that, that roughly correspond to these 2 phases. So they took these 32 drug classes, things like statins that lower cholesterol, beta blockers that are useful for treating blood pressure, drugs that are useful as antidepressants, 32 classes of them. And they said, did public sector contribute to these areas, or did the private sector contribute to these? And it doesn't show up. In point of fact, what their data showed is that the public sector, universities and other public sectors, heavily contribute to the basic science, and the private sectors heavily contribute to the other stages. And so, in fact, uniquely, particularly in the US system, is that the public sector and the pharmaceutical research efforts are completely complementary. The former weighed more heavily towards the basic science, and the latter more heavily towards the discovery, synthesis, and testing of the compounds. And what's unique about the system that's been developed in the US is this hand-in-glove complementarity of the public and private sector to achieve medical benefit for all of us. So having said all that, this is another depiction of what I've showed you in terms of discovery and development. How long does it take and how much does it cost? Up here is years for each of those phases. So imagine that you're just coming out of school with your PhD. You might be sort of 27, 28, 29, 30, somewhere in there. You start working on a project. If you're 30, by the time you're 43, if you're lucky, you'll see your first drug. Oh, and by the way, along the way, you will have gone through about $850 million. Not you personally, but the project will have gone through about $850. That splits out between, if you look at these 2 points in which government regulatory agencies' approval is required, the investigational new drug or the new drug application, takes about 7 years and about $600 million to get to this point, about 5.5 years and about another $230 million to get to this point. This was one study, a separate study using a different set of data said about 5 years. So you can see they're pretty comparable. In fact, that's a little bit misleading because that actually includes all of the failures at this stage of chemicals that don't even get to the government regulation. In fact, if you look at those, the failures of the 620 million, about 500 million is spent on failures. So if you could just double, not even double, go up 1.5 times more effectively at this stage, you could remove millions of dollars that are, in a sense, wasted from the healthcare system simply by incremental improvements at this very, very early stage. These costs have to be recouped because they are good ideas that fell somewhere along the way, be it efficacy or toxicity or optimization or invalid target. In fact, you can look and see over a decade why drugs fail. If something's been commercialized but later withdrawn, not surprisingly, 94% of the time, that's because they have some toxic effect when they get into these large patient populations. If they're terminated in those early phase 3 trials, about 3/4 of the time, it's between one of two things. It's either toxic or it flat out didn't work. So again, minimizing toxicity and improving efficacy would have a huge impact on how rapidly drugs are developed and how much they cost during the development cycle. In fact, you can even dig down further if you're really interested in why those phase 3 trials fail. About half the time they fail because they're no better than placebo. You may as well take a sugar pill as spend money on the drug. About half of the time that they fail is because their safety profile is unacceptable versus placebo. Either you saw an early safety signal that was confirmed, or there was just some safety issue that cropped up and you're not quite sure why. And finally, there's some that fail at the phase 3 because they're really no different than something that's already out there. They're no more effective and they might not even be as safe as what's out there. And so the drug is just stopped. So if you look at modern drug discovery on expenditures, and this stops in 2004, but it's continued to grow. In 2004, there was about $40 billion spent in drug research and development. This is expressed in constant 2004 dollars. So that's a growth from 1994 of about 17 million to about 40 million in 2004. This line shows the total number of those submissions to the regulatory, in this case the US FDA, new drug applications that were submitted. And it's very hard to think that that line is anything but completely flat. In fact, it gets even worse if you eliminate from this line all of those drugs that are so-called Me Too drugs that look kind of like something that's already out there, and just look at new medical entity submissions. That line is completely flat. Despite a massive increase in expenditure, new drug approvals, new medical entity submissions have essentially remained static for over a decade.
Despite a huge increase in investment.
Despite the huge increase in investment. So in fact, if you portrayed that figure a different way, which is dollars per drug approved, it's just massive because expenditure has gone from 17 billion to 40 billion. New medical entity submissions are pretty static around 5, 8, 10 per year. So the reason always is why, and that in fact is where we think genomics can have a huge impact. We think it's because targets, there's a number of reasons that people have hypothesized. Some people might say, well, all the easy targets are taken. They've all been worked on already, so all we're left with are the really hard ones. Some people might say, well, if you have a disease like hypertension or diabetes, those are easy to measure, those pharmacodynamic endpoints that I talked about. Blood pressure is a dynamic endpoint. You actually, it's not that you want to lower your blood pressure. You don't want to have a stroke or a heart attack, or that's why you want your blood pressure down. But it's a surrogate. It's a dynamic endpoint that you can look at. Blood glucose is the same thing. You want to lower your blood glucose so that something else doesn't happen. And some people might say, well, all of those so-called easy diseases have effective therapy, so we're being left with harder diseases like Alzheimer's disease and cancer. Some people might say that the amount of regulatory requirements have risen so high that the cost has gone up because regulation has added additional layers of bureaucracy and maybe not even bureaucracy, appropriate safety levels, and so the cost has gone up. As many reasons as you can think of, people have conjectured. The bottom line is that's the reality, and many people are trying to think of ways to improve it. I've tried to highlight for you as we went through some of the places where maybe by understanding more about genetics, you can get better validated targets, you can use robot and engineering efficiently to identify compounds, you can target patient populations more precisely. But in fact, that's where the opportunities are to transform medicine. At the Broad, it started with the Human Genome Project and genome sequencing, moving through genomic tools that tell us differences in the genetic composition from individuals to individuals, an increased understanding of the biological basis of disease, and now we're ready, we think, to build on these genomic discoveries that started about a decade or more ago, to begin to say how can we truly make the process of drug discovery novel by building on these genetic tools that have been and understandings that have been gained over the last decade, and that if we can do that, we will in fact transform medicine. We intend to do that here by sort of turning the traditional process that I've described for you in some, maybe some of you are thinking painful detail, a little bit on its head. Traditionally, you start with that protein in the test tube. You start with a target, you go through the screening, you optimize it, you develop the scale up, you scale up, finally you test and say, does it do something useful on a cell or an animal or a human? By understanding the genetics, we actually think we can begin by testing in vivo chemicals that achieve an effect we want at the end of the day, and then do the optimization through an understanding of both modern chemistry as well as modern genetics. Hopefully, getting through this process more efficiently, more precisely, in a target-directed fashion. Whoops, that got out of place. So we're finally at the end. I've told you what's a drug. I've told you some of the stages of drug discovery and development, some of the costs and risks of drug discovery, and some of the changes that we think we can make, both in the traditional process as well as exploiting some of the human genetics. I thank you for your attention. I apologize for running a little late. Be more than happy to take a few questions. But again, thank you for your time. Yeah, here in the front row. Please.
I was thrilled that you focused on statins so much because I'm on them. And in the last month, I have been troubled by the fact that there are several controversies about the appropriateness of statins for certain populations. Of course, I guess we've all read in the last couple of weeks the controversy about the proposal to give more statins to children,
Right.
or to give statins to children at all. And the other controversy is that there's several popular books about medicine that claim that people like me who are over 70 and have no background personally of heart difficulties, that mortality is not really affected by being put on statins. So there are these two controversies that I've become aware of in the last month about statins. I'm wondering if you care to comment on either of them.
I'll comment, but I'll preface my comments by saying I am not a physician. I have never gone to medical school. I'm a PhD in biochemistry, and so everything I say should be taken as my opinion. And you should check with your physician before you go off of your statins at all.
You won't
So that's my disclaimer. Both of those questions actually come back to the efficacy benefit, risk benefit, efficacy toxicity ratio that I've been talking about. So the question really comes down to is, in children, is the benefit of lowering their cholesterol early outweighed or balanced by the potential risk of exposing them to that chemical earlier rather than later. And that is a controversial question, and it's because you are weighing, and it's the same whether you're at that end or the over 70 end, it's actually the same question. Is the risk, any potential risk worth the potential benefit? You know, with cholesterol, particularly at the older end of the spectrum, and particularly from the Framingham study, it's pretty clear that elevated cholesterol, even without a family history, is associated, in a population basis, is associated with cardiac risk. And I think that's the driver for people saying, well, we should keep our cholesterol down. The pediatric population is a difficult one. So, yeah.
I was wondering if you could comment on the importance of computer scientists and drug discovery, and at what stages they come in handy.
Sure. So a little bit of computer science is what you mean by that. So, but I'll just describe places at which software engineers and other kind of people, I'll just describe various places, and you can, if one of them fit you, good. So they play an important role at the early stages when you have a biochemical target and you might have a crystal structure of a protein, and you want to ask questions like, can I take a small molecule and can I, in silico, in the computer, model how that small molecule is interacting with a protein of interest? Because that can tell me how to design a better molecule. We will next year generate in our upstairs, in our high throughput screening robotic system, we'll generate on the order of 15 to 20 million points of data that will be associated with hundreds of thousands of compounds. We have to be able to retrieve, track, find that data, associate that data, and so that we actually have a fairly intensive group working in informatics to just help us build the infrastructure of tracking that data, mapping that data, knowing where the data is, creating associations that say, oh, here's a chemical that affects this process, here's a chemical that affects that process. Those processes are related. Gee, maybe we can pull that data together. A human being cannot handle 20 million data points in their head at all. They play a role in designing, in modeling statistical outcomes of clinical trials. So there's various, the simplest clinical trial design is one that says, here's a group of people that are going to get a sugar pill, here's a group of people that are going to get a drug, I'm going to compare this group to that group. That's pretty much the simplest trial design. As you get into more sophisticated trial designs in which you might say, well, I'm going to start a person on a little dose and then if I see an effect, start to move them to another dose. Mimicking that kind of outcomes is an important component for sort of statistical analyses. Analyzing, playing a role in analyzing patient populations to ensure that you've not unintentionally introduced a bias into your studies. So for instance, if you were doing a study in which, let's use the simple trial design, sugar pill drug, but by bad luck you had a preponderance of women in this group and a preponderance of men in that group, you would have an invalid study, and you would need to model that as you were going through to say, gee, we're getting an imbalance in our groups. So in fact, all the way through the process, we have become virtually completely dependent upon computer scientists to help us develop the tools that we need in the process. Let's go over here.
What's the ratio of significant drug discovery and development in the United States compared to the rest of the world, and how's that changing?
The U.S. is by far the 5 gazillion pound gorilla in the room. It dominates in the just in sort of a qualitative sense. In terms of a quantitative sense, it's as I recall. So I'm relying on my often imprecise memory. It's about a 5-to-1 ratio. What's changing over time is that more and more of the remember I talked about this lead optimization, designing chemicals? More and more of that activity is being outsourced to particularly China and India, in which there are highly trained populations that are able to do that kind of optimization. To date, at least, much of that is being done in which scientists in the US do the design work as to what compounds should be made, while the actual execution of the design is being offshored or outsourced to other economies. And so it in fact is a very dynamic environment right now with things changing very, very quickly. If you pay attention, you may have seen companies like an Indian company called Ranbaxy that's been buying up a variety of drug discovery companies around the world to increase their activity in the early drug discovery stages. So it's going to look very different in the next years than it does right now. And it certainly looks different now than it did 5 years ago. Oh, gosh. Let me take, I've got so many questions. Let's do this. Let me take 3 more, and then I'll be happy to take any more afterwards. So I'm going to do this completely randomly. If your birthday is January to June, keep your hand up. All right? If your birthday is, no, don't stand up. If your birth date is between the 1st and the 15th of the month, keep your hand up. Oh, gosh. How many do we still have? We have 1, 2, 3. We have 4. We can do 4.
Let's adjourn. We'll start the reception. And if you guys have additional questions, you can come down and Robert will answer them. How's that sound? Okay. Thank you so much.